think about against next week. So what we're going to be doing today, see if it goes right, okay, is look at the way in which we handle bacteria in the lab. So how we grow them, culture them, how we work with bacteria essentially. And we're also going to look at how we prevent bacterial growth. And so what we did last time, kind of introduce, what we're going to do with our discussion on pathogenesis and diseases. So looked at, caries, dermatitis, and as well. And so when we talk about those in detail, we're going to find ourselves talking about isolating cells, trying to culture them, trying to make decisions if that should be done. And how do we identify them if we do grow them or culture them, and then also how do we prevent growth. And so everything from physical means, chemical means, etc. we'll talk about, to see how it is that we do that. And so before we get into that section, we want to finish up what we had left yesterday, which was to compare exotoxins and endotoxins, just so we really have the properties of the two types of toxins, and so that we can answer questions about them as well. So we had stopped right here. We had talked about the way in which endodontic infections are kind of thought about, and we made the point that unlike the other two conditions here, we're essentially dealing with things the way we would do wounds, okay, because we don't have a whole set of organisms that we would identify being in here, although there are one or two that kind of typify these type of infections. And so we're going to look at that. But so when you have this type of situation where you have the polymicrobial microbial infections, as much as possible, when you're trying to isolate organisms and evaluate what's there, we have to maintain a septic technique. We have to decide if we're going to culture or not culture, if we're just going to treat and prevent and do all of that without really knowing the organisms that are present in the infection. And to do all of that, we have to maintain sterile conditions as we're working, so that we don't contaminate sites and so on with additional bacteria that may or may not be important in those infections. And so again, before we look at topics for today, we want to make sure we understand the differences between exotoxins, endotoxins. So we mentioned endotoxins, we talked about LPS. We know the three parts of LPS, the lipidae, the core, polysaccharide, the O, antigen, and so on. And we mentioned that exotoxins, we typically think of gram-positive cells when we see exotoxins. But we also want to keep in mind that gram-negative cells can also produce exotoxins, even though we predominantly think of gram-positive cells. And so when we think of exotoxins, we're thinking about molecules that are soluble, heat labile, the proteinaceous, so macromolecule-wise, proteins are what we think about. And so some of the toxins that have had, really important contributions, if you will, to the field of bacteriology and public health have been, some of the ones that toxoids could be made from, following a heating of them and so on. And so, toxins altogether, produced by bacteria have a very important place in microbiology and by extension in public health because of the things that can be done with them and the damage that they can cause. And so exotoxins are secreted into surroundings as the pathogen grows. So remember, when the bacteria die, right? We have the LPS released, and we have the different effects that we already mentioned. And so again, most of the exotoxin producers are gram-positive, and this includes both gram-positive rods as well as gram-positive cocci. So it's not limited to one or the other. And so when the exotoxins are produced, they can, move or travel from the site of infection to other locations in the body, to other tissues, and, in many cases, they can interact with molecules on the surface of the cells, and they can mediate their actions that way. And so we have, many different types of exotoxins, as we'll see. And so this next point is pretty important. So, the bacteria that secrete the exotoxins, are pretty specific, and we can always associate the toxin with the bacteria. And so we have exotoxins produced by, Clostridium, by Bacillus, we associate them directly with those organisms. But what's important is that in addition to having the toxins encoded by genes on the bacterial DNA, we can also have plasmids as well as phage encode the toxins. So when that happens, and in our discussion of biofilms, right, we can have the plasmids exchanged, and we can increase virulence of the organisms by now having them receive these plasmids, with the DNA in there that can encode for the toxins. And the exotoxins, again, they're very specific in action sometimes. Others have a more broad effect, but they're lethal. Okay, small amounts, of toxins, can be lethal. So, in the category of things, that are cytotoxic, things, that can kill cells, the exotoxins, rank very high amongst the most lethal substances that are known. And so we see some examples of this. And the highly immunogenic, and as I mentioned a few minutes ago, the toxoids that are produced, when the toxins, are heated, have been very important, in terms of, vaccine use and things of that nature. So they're highly immunogenic, they can stimulate an immune response, we can have a strong adaptive immune response produced against them, have, antibodies with high titers, made against these molecules. And so the antibodies that are produced can have antitoxin, effects, they can help to neutralize, the organisms, because of the toxins that are now bound. And again, some of the really important ones, as mentioned over here in examples, we can have chemical inactivation, we can have human activation. But one of the best examples of the toxoid is the tetanus toxoid. Okay, and so again, this is inactivated. The immunogenicity is still there. So in other words, it can still elicit immune response, but the active component of the toxin, okay, is no longer in effect. So things that we need to remember about this. So there are different types of exotoxins. Some of the ones that have gained a lot of interest, are these ones that are referred to as the AB exotoxins. And what we need to remember, for our purposes, for the class here, is that the two subunits, right? The A subunit, is usually the one that has the active component. And then the B subunit is the one that will interact with the target cell. And there are many permutations of these AB toxins. So one of the toxins that has been studied a lot is the toxin from Vibrio cholera. So we're all kind of remembering that when individuals have cholera, one of the things that happens is this profuse diarrhea that that results, where you have a lot of water loss. And so in that situation, we're dealing with, the A components, we have 5 B components, and they interact together, and, once the target cells are bound, in the epithelium, we have this cascade of events that are released and activated within the cell, where we have, the cyclic AMP components, generated, we activate, protein kinase A down the way in the pathway, and we, phosphorylate, chloride channels on the epithelial cells, and begin this, whole process, of having these ions rush out and water rush out following, which is why we have the watery diarrhea. And the characteristic of that particular diarrhea, because of the destruction that's also going on in the epithelium, is this, very flecky kind of stool that's referred to as the rice water stool. And so we have the characteristics of that over there. So again, these AV toxins, are pretty important because of this, if you will, division of labor that's taking place, where the A component, the A subunit is doing, the activation, and then the B component or components involved, with the binding to the particular target cell, that the toxin is aimed at. And so again, we have, specific host site, for these exotoxins. We have ones that can disrupt membranes, and then, a group that's categorized under the exotoxins are the superantigens. And so again, I don't know how well you can see these figures over here, but you can't see them very well. When you look up close later on, you can see the different things in there. But pretty much, you're being shown, what happens when, we have the binding, of the B component to the cell, the uptake, of it, and then the events that occur within the cell. So for us, the main thing we want to remember is, what does the A component do? What does the B component do? Right? We want to remember activity with the A, the binding, with the B. Okay. And then, pretty much, down here you're showing what can happen, so that, if we, affect, the membrane of the cell, once we've gone in, so we can disrupt physiological aspects of the cell, and we can move water and ions and things of that nature across the membrane. And so, go ahead.
With the A and B exotoxin, is
Right.
the A like a type of enzyme that disrupts?
Some of them have enzymatic activity, they're catalytic. So, and that would, the activity will be realized once the B component, has interacted with the target cell. Yeah, yeah. So this table over here, again, just kind of gives you examples. And when you look on the left over there, right, there are toxins that we're familiar with. So we've mentioned the cholera toxin, which is the one, down here. And, again, mentioning that, we have the cyclic AMP that's generated, and, in huge amounts. So if you remember your signaling, what happens when you have the cyclase, activated, and it cleaves, the ATP, we generate that cyclic AMP, right? We act as a second messenger, and we can do, signaling under very nice regulated conditions. But with this particular situation, the cyclic AMP levels are so high that, again, we lead to these kinases, one of them being protein kinase A that's, activated, and then we phosphorylate the channel, and then we have all this stuff going on. So again, when you're looking up here, we want to recognize, the ones that are the ABs, and, and we have their effects, kind of different from the general cytotoxins that will disrupt cell activity. So the differentiation between, the ABs and the cytotoxins is the fact that, the ABs basically affect internal activities within the cell. Okay, so in this case, like the signaling pathways. But then you have other things in here as well. So, we mentioned the superantigens. And so if you remember, when antigens are being recognized by T cells, right? T cells, with the T cell receptor will recognize the epitope on the antigen, and then the MHC molecule, right, is also combined with the antigen in the non-T cell recognizing portion of the antigen. And so when binding takes place, right, either CD4 or CD8 will interact with the MHC, right? The T cell receptor interacts with the epitope on the antigen. The T cell, gets activated, okay, as a result of that interaction, and we disengage, okay? So here with the superantigens, right, that doesn't happen. The antigen is bound, to the V beta domain, okay, of the MHC, and as well on the T cell, and it stays there. And so the T cell just keeps getting activated and producing all the cytokines, and all those cytokines that are being produced, have their downstream effects. And, we can just have this, exaggerated response that takes place. And so, it can lead to cell death, it can lead to organ damage, it can actually even lead to death of the host. And so, the activity that we have from the superantigens, and again, different bacteria produced in different types, makes them, be placed into this category of the exotoxins, because again, the things that the bacteria are producing that are leading to this kind of an effect. And so, we see, what I've just described over here, so that you can see, the MHC molecule, the superantigen is associated, with that on the APC. You see that on the V beta region of the T cell receptor, and so we have this, hold, that just stays there, and there's no disengagement, and so we have this, continuous activation of the T cell and the various cytokines that are produced. And so when this happens, again, we can have a lot of, things happen. So multiple organ failure will occur. This huge cytokine release, will cause all this damage. And so we have the staphylococcal enterotoxin B, that can initiate this kind of a response. And so we want to kind of remember this aspect of things. Okay, so we are talking about the superantigens and how it is, that they're affecting the cells. So we have an example over here to look at. And this is one of the earlier ones that was, that was studied quite a bit because of things like toxic shock syndrome that, that would result in a few cases that would resulted in death. And so we have the endotoxins. So again, this is LPS, gram-negative cell. We want to remember that the component on the LPS, that has the effects is the lipid A. So again, we want to remember the three components of the LPS. So the lipid A is the one that has the toxic, properties, and that's the one that elicits all these reactions that we've talked about, everything from the mitogenic effects, to stimulating, inflammatory type reactions, from the immune system, and so on. So when you see descriptions of characteristics or properties of an exotoxin, we want to be able to differentiate that from the endotoxin. Does that make sense? Okay, so that's what we're trying to do. We're trying to kind of get that, fundamental difference between the two. Now, we have the designation endotoxin, right? Because it's the endogenous part. So remember, your O antigen sticks out on the outside, core is in the middle. The lipid A we have embedded into the outer membrane. Everybody remember that kind of picture visually? Okay. And so, the majority of what's produced is when the cells die. A little bit, can be produced as the cell is undergoing division. And we'll see when we look at the systemic, infections and so on. I think that's the last Monday we're here. We will see how important this is. Okay, so just to remember that core thing. So we've seen dichotomous charts, yesterday. How we can go through and differentiate between, gram-positive, gram-negative, and within the gram-positive, gram-negative, the different genus and species. We talked about yesterday. And so here, we're just kind of looking at the various effects that we can have, from endotoxins. So when we look at systemic, infections that last week, right, we will look at this again. And so what we're trying to understand at that point is that, when we have the LPS, that lipid A available, right, we can set up, various cascades. So pro-inflammatory type cascades, that deal with the kininogen, fibrinolytic type complements, coagulation. And then we have all these mediators, in the blue box in the middle there. And so, what we want to see is, the effects that they have. So on the cardiovascular system, we have that on the vasculature itself, multiple organs, and then individuals, will go into a shock, and then we have, multi-organ failure, and so on. And so, if there's intervention, at a suitable time point, quick enough, there can be a recovery. Otherwise, it will lead to death. So LPS, is of great concern, okay, because of these things that we see over here, okay? And so when we look at systemic infection, and if we have organisms that have, you know, left the oral cavity and then now in the blood, and then, you know, we have them, you know, in places like the heart, for example, you know, around the heart valve and so on, if the organisms, you know, die and we have all this LPS released, then, you know, potentially, you know, we're concerned about the things that we see over here on this slide. And so, unlike the exotoxins, right, which we said, you know, were heat labile, so here, you know, the endotoxin is heat stable, you know, nanogram amounts, you know, toxic, and the immunogenicity, you know, compared to the exotoxins, you know, is weak, and so we don't have, you know, much heterogeneity, right? Because again, you know, we have just that lipid A portion, you know, the lipidic component, right? The O antigen, you know, which is the one exposed on the surface, has a little more variation on it, right? But the lipid A, you know, is pretty much the same, okay? You know, from E. coli to Shigella to Salmonella and so on. And as we saw, you know, in the figure just, you know, previously in the previous slide there, right? You know, we see these various components over here that can be the result of having the LPS, you know, available. In particular, the lipid A component. So everything, you know, from, you know, fever, weakness, etc., inflammatory responses, all the way to the multi-organ, you know, type of thing that we saw on the slide before. So we want to kind of see what is involved over here. So the endotoxins, right? So they can interact, you know, with cells, you know, with molecules to do the activation we saw in that figure. And so when we have them interact, you know, with macrophages, you know, take these up. We can release pyrogens, which is what induces the fever. And, you know, when we bind to LPS binding proteins, again, remember, we have toll-like receptors that can bind to the LPS, right? We can lead to the release of cytokines, and most importantly, pro-inflammatory cytokines. And so things like IL-1, okay, so things like TNF-alpha, right? And these are the things that will lead to the septic shock that we saw further down in that figure. And so just to contrast things, you know, we don't have to do anything, you know, with this. You're going to be asked any question about it, is to just to say that we have other organisms that produce toxins, and the fungal types and the mycotoxins. We have, even amongst protozoans, you know, that have GPI anchors, you know, to the membranes, you know, when those are shed, they pretty much have, you know, toxic effects and will elicit, you know, immune responses and so on. So this is not something that's unique to bacteria, okay? So, you know, we have in fungi, we have in protozoans and so on, and these can have, you know, important effects and all of that. And so some of the ones that I'm most familiar with are the ones that we have, you know, in grain crop, for example, with aspergillus, so on, aflatoxins. So these are all examples of mycotoxins. And so we just kind of wanted to remember that. So the toxic component of LPS is which of the four?
Lipid
The lipid A. Okay, so lipid A. So if you're shown, you know, a diagram of LPS, and you have, you know, an arrow pointing to, you know, portions of it, you know, can you recognize which is the lipid A, which is the O antigen, which is the core? Okay, and then of course, you know, to be able to relate it to these different effects that we have. Okay, so we talked about bacterial endospores before, and we made the point yesterday that when the endospore forms, everything that the cell is going to need to develop vegetatively is present in that spore. And that is why it becomes important to eliminate that spore, to kill it, okay, when we're trying to sterilize, you know, media and instruments and whatever it is that we're autoclaving. It's important to get rid of those spores. And so here we have, you know, the spores allow the bacteria to survive, okay, you know, in the environment when, you know, nutrient levels are low, environmental conditions, you know, not so great for growth. And then once, you know, we have, you know, nutrients available again, you know, we're no longer under adverse effects, then we vegetate, right? So the bacterial endospores, they're resistant to the environment, and adverse conditions because of the spore coat, the exosporium, and what else? What else on this list? I hear two things. Which one is it? A. Okay. So we have the glycolic acid, with the calcium complex, and this helps to impart this high resistance to the spore. And so when we need to kill the spore, just heating, for an hour is not going to be enough, right? We need to penetrate, this, spore coat. We need to get inside to kill the vegetative cell, as we're going to see. We need, moisture and the heat and pressure, okay, to be able to do it. Okay. So the things that we want to look at today, right? So cultivation methods, how do we grow cells? What kind of nutritional and environmental factors do we need for the growth? And the factors that affect growth, both aerobic and anaerobic growth, and the kinds of media and inoculation methods that are used. And so, we would not be complete if we don't at least look at this, just so that when we're talking about decisions to either culture or not culture later on, and how we're going to identify things, we have, in the back of our minds, what differential media is, what selective media is, what you're plating the cells on, how do you, quantitate, how do you enumerate, the cells that you have to kind of make sense of what you're dealing with. And so, cells have all kinds of nutritional requirements. And so, some of the basic things, the requirements for carbon, nitrogen, phosphorus, sulfur, etc., oxygen, if they're aerobic, are pretty important. And the cells will also need a variety of micronutrients, for their growth. And so when the media is made, right, we have media that's defined, in which case you actually know exactly what you're putting in there, and then we have the media that's not defined, where we know components that are required for growth are present, but every time we obtain that nutrient source, there might be differences in the amounts of the components that are present, okay? So when we talk about something like nutrient agar, everybody's heard that term before, nutrient agar, right? You have nutrient broth, you're gonna grow these cells in it, right? So, we have different extracts that are part of the media in there that we use every time we prepare it, but we don't know exactly, how much of each component is there, right? But it works, and so, we use it. And so in defined media, we know exactly what's in there. So we have the requirements, for minerals, for vitamins, as well as the macromolecular components that are needed so that the cells can metabolize. And so, when we have cell, go ahead.
Was the defined, both the components and the amounts, and then
Yes.
the undefined?
Yeah. So in defined media, we know, so basically you're measuring everything out, you know exactly what's in there, right? And so, and then we'll see as we get to, the examples, here in a minute.
So
Yeah.
on undefined media, do you still know the components, just not the amounts, or you don't?
Well, yeah, so, you can always measure, 10 grams of the same thing every time you're making the media, but, if you have, yeast extract in there, you don't know how much of all the components you have in the extract that are there every time. But, there's enough, minimally to allow the cells to grow, okay? Does that make sense? Right, so when we're working with defined media, especially with minimal media, okay, every single time I'm gonna weigh out, 2 grams of dextrose. I'm gonna put in, 0.5 grams of calcium. You know exactly what it is in there, okay? So there's certain times when that requirement is absolutely necessary, yeah. Okay, and so over here, we just wanna remind ourselves that we have, the 2 big categories, for nutrition requirements. We have autotrophs, we have heterotrophs. This is what you would need to remember. The subdivisions underneath that, we're just going to talk about them, but, we don't have to, I'm not going to belabor the point. So we have, in terms of the autotrophs, these are the ones that, they get the carbon from inorganic sources. So we have, cells that are photosynthetic. right? So they can photosynthesize, and they can, you know, produce carbon products that are stored. And the heterotrophs, you know, depend on the autotrophs. And so they will, you know, obtain their carbon in pre-existing organic forms. So if we, you know, we can further, you know, break them down into those that are requiring, you know, the chemical sources, which is those, again, that are requiring, you know, light for that. And we can look at, you know, what they need for electron sources and so on. So we can define, you know, the different bacteria into much tighter and narrower categories based on their nutritional requirements. But what we want to remember are just these two big groups, the autotrophs, the heterotrophs, okay? Nitrogen is very important, you know, as a nutrient source. And, you know, in many instances, you know, the nitrogen cannot be used as it is. So cells will obtain, you know, the nitrogen from other sources, you know, things like amino acids, nucleic acids, and then they need to be able to, you know, have the nitrogen available in usable form. And so again, you know, this is just showing an example of that. And so even, you know, in our cells and metabolic activity, we need nitrogen. You know, we cannot use the nitrogen as it is available in the atmosphere. We have to have it, you know, fixed, you know, so that the plants can produce nutrients, and then we get those nutrients from the plants, and indirectly we get the nitrogen that way. So the bacteria are no different in that respect. And so what we have, you know, bacteria that can actually fix the nitrogen. We have ones, you know, that can allow the usable forms to be made, and they can also extract the nitrogen back and put it back into the atmosphere. So again, this is just a reminder of the need for nitrogen. And so for energy use, you know, the categories that we saw, we have the phototrophs, they capture light energy, they'll make ATP from that. The chemotropes, you know, they will do oxidation, you know, get organic materials reduced. They can also use inorganic compounds for that, and so they can still metabolize either way. You know, we have bacteria that can, you know, basically extract the energy sources, you know, from things like rocks, for example. They can get, you know, electrons from there. So we have different, you know, ways of being able to obtain nutrients, you know, by bacteria. And so when we look at, you know, what the cells, you know, need, again, minimally, right? We need to first think of where is the organism living. So if we have organisms, you know, that are aerobic, their nutritional needs are going to be very different from organisms, you know, that are anaerobic. And so the examples, you know, also extend over here. We have, you know, organisms that are phototrophic, they can synthesize what they need. If they're auxotrophs, you know, if they cannot do that, they're going to need precursors, and then they can generate the carbon sources. And so we can measure, you know, the growth of cells. So again, if you're looking at this, depending on where you have the cells growing, right, you can have ones that, you know, under very high nutrition concentrations, we can have, you know, huge amounts of growth. So we can measure, you know, by taking optical density readings or OD readings, we can see how quickly the cells are growing. We can plate the cells out, you know, and look at, you know, colonies that are produced, and we can count those and get a sense, you know, of just how well things are growing. And so a lot of the growth rate will depend on, you know, the nutrients that are available. So again, you know, medium nutrient concentration, you know, in between, low nutrient concentration in between, and so we're measuring growth, you know, over a period of time. And so if you remember that bacteria can divide very, very, very fast, okay? So, you know, on the average, you know, you have cells that will divide, you know, every 20, 30 minutes. E. coli, you know, as 15 minutes. And so if we're measuring your growth, you know, we're doing that over a period of time. And so when you look at the nutrient needs that cells will have, right, the second point, you know, becomes pretty important. So you're going to have, you know, one key nutrient that's available in the lowest amount will dictate how much the cells will grow over a period of time. And so when we look at biofilms, again, you know, later on, you know, we want to keep this in mind. Okay, in terms of, you know, what group of organisms can dominate, you know, within the biofilm. Okay, so again, you know, you have a limiting factor that can, you know, determine that cell growth. And that's usually the key, to determine why those cells are predominant. So aerobic organisms need oxygen. Anaerobic organisms, cannot grow, when oxygen is present. And so amongst the aerobes and anaerobes, we have obligate aerobes must have oxygen, obligate anaerobes, no oxygen allowed for growth. And then we have grades in between. So we have facultative organisms, facultatively aerobic, facultatively anaerobic, and we're going to encounter those, in the oral cavity. And so we have microaerophils. They will grow best when there's less oxygen than normal. So but you have a gradient. And we have aerotolerant anaerobes. So if oxygen is present, they will not die from it. Okay, but they will not utilize it. And so the facultative anaerobes, they can use oxygen, but also if oxygen is not present, they can still grow. And so a lot of this, will affect the metabolic activity of the cell. So if the anaerobic, we would not expect, them to have, oxidative phosphorylation going on. Okay, everybody kind of understands that. So if we have the aerobic, then, that's going to be part of the metabolic profile. They'll be able to undergo oxidative phosphorylation. We need, oxygen, as the electron acceptor. But because we have varied ways that the cells can grow, right? Not every cell is going to use oxygen as the terminal electron acceptor in oxidative phosphorylation. And so we just, seeing over here what happens when you have oxygen, for microbial growth. And so cells will produce, a number of substances to help mitigate the effects that you can have from free radicals that are produced as a result of oxygen use. And so you see, the examples of the radicals that are produced. So, the single oxygen species, peroxide anions, hydroxyl radicals, etc., hydrogen peroxide. And then the cells have, essentially defense mechanisms to prevent, damage, from those. And many of these are antioxidants that can, help to prevent, these radicals from essentially associating, with DNA in particular, right, and being able to destroy the other cell membrane components that can actually end up damaging the cell. So you see the examples for each case, right? And at the bottom over there, we have hydrogen peroxide, we have catalase and peroxidase enzymes. And so on the chart that we looked at, the dichotomous key chart, and we saw, the cells that were producing catalase, right? So that is an important component because again, it helps to prevent damage, from the peroxides that are produced. And so that's a very key characteristic of those cells that do that. And so if you put hydrogen peroxide on, on those cells, you see them bubble, and that usually tells you that, that they have the catalase that's being produced. pH, effects can be important. We need to know the difference between what we're seeing over here, the acidophiles, neutrophiles, alkalophiles. And so, we have cells that can grow at different, pH ranges. And so, you see, the pH levels, 9 and above. The neutrophiles right in the middle, most of the organisms will fall in there, the general type of organisms that we would encounter. And then we have these acidophiles, that grow at pHs, less than 6. And so we need to remember these terms right here and the pH ranges right here. So the pH, of media and environment, within tissues where you have bacteria growing is important because the pH changes can affect, protein, can affect, enzyme activity. It can affect, nucleic acids, etc. It can, also affect transport of molecules, across the membrane, with electrochemical gradients, and it can affect cell growth. Okay, so cells, that are growing outside, if you put them outside their range, right, can get damaged and they will not grow. So we need to know these terms and the pH ranges that we have the cells growing. Other things that are important, and we're just going to mention them, you know, in passing real quick on this slide: osmotic pressure, the availability of water for growth. And so again, you know, the solid concentrations you have, whether things, you know, hypotonic, you know, hypertonic, you know, isotonic, will affect the way in which the cells, you know, will grow. And so if, you know, if you have a cell, you know, you put it, you know, in a hypertonic environment, you know, you can have water loss, the cells can shrink. If you have them, you know, in a hypotonic environment, water can go in, you know, cells can swell, you know, and burst. So the conditions, you know, tonicity is important, you know, for cell growth. And so, you know, also realizing the fact that if the cells are going to carry out, you know, the various metabolic reactions, right, water is needed, and so that becomes a very important component for ensuring that cells will grow. Temperature is also important. So again, we have different ranges. So just like with the pH, you know, we want to know those terms over there. So we want to know, you know, the thermophils, the mesophils, cyclophils, and the hypothermophils. And so in this group over here, you see, you know, the three domains. We see the eukarya, we see the archaea, and then we see, you know, the bacteria. And of the three domains, the one that can live, you know, in the most extreme environments are the archaea. So extreme in terms of temperature, in terms of pH, in terms of osmotic conditions, I mean, you name it, right? Very extreme. You have cells that can, you know, survive in those environments amongst the archaea. So we need to know these terms to be able to recognize them. Okay? And so when we grow cells, okay, in the lab, okay, so the typical, you know, type of media, you know, that would be used, right? Solid media like we have with the agar plates, or liquid media and broth. Okay, so we have broth, you know, media, we have solid media on the plates. And so pretty much, you know, what this is showing you over here is that, you know, the agar component that's mixed in, you know, with the broth media to make the solid media, right, you know, is powdery, dried material that, you know, you basically weigh out and you add to, you know, to water. You hydrate it, okay, and then you put the components either already, you know, mixed together, your nutrient broth into that mixture, and then you melt the agar, and then, you know, you autoclave it, and then you pour your plates. And so the plates now, you know, once they solidify, can be used, you know, to grow the cells. And so you can either streak directly on them, you can either pour, you know, do pour plates and put the cell, get the cells onto the surface of the agar, and you can incubate them to get, you know, the cell growth. And so when the cells grow, you're looking for the colonies, okay, that are formed as a result, you know, of the growth. And so in the way that we think about things, you know, is that if we have one colony, we're thinking that that, you know, came about as a result of, you know, one single cell, okay? And following division of the cell and accumulating the number of, you know, bacteria, we now get those colonies that we can see, you know, visibly, because the bacteria, of course, we can't see, but we can see, you know, the evidence of their growth as the colonies, and in the liquid media as turbidity, okay, which we can measure in the spectrophotometer. And so this is showing examples, you know, of the different, you know, media and the appearance of cells on the media. So what you're seeing are different types of microbial growth, different types of colonies. And some of those colonies are highly characteristic of a given organism type. So when we're talking about, you know, something like Porphyromonas, you know, gingivalis, and we have these, you know, black pigmented colonies, whatever, this is characteristic, okay? And so you're seeing, you know, these circular colonies, you know, they're pretty small, right? The compact circular type, we have filamentous type growth, and then we can have different textures. The colonies can be dry, they can be smooth, they can be raised, and so on, each one telling you something about the type of cell. And as well, you know, some of them have characteristic odors to them, so that, you know, when they're growing, you know, and you can smell that, and you know, okay, these particular organisms, you know, are in here growing. And so we see these examples. So we had you know, a little bit of a discussion about, you know, the types of the media. So we can have complex, okay? And so here, you know, like the nutrient agar example we just saw, we don't know the precise chemical composition of everything that's in that media. You have extracts from various sources, you know, that we know have protein, they have minerals, they have vitamins, they have all the different things, you know, the amount of, you know, carbohydrates and so on that the cells will need, but we don't know exactly, you know, what they are. They can change from you know, production to production, okay? So when we have that, those are complex media, unknown chemical composition. Or we can have the defined media, as we said, where we know exactly what's in there. We can have minimal, you know, media, minimal essential media, where, you know, again, you know, synthetic components, you know, stuff off the shelf, you know, you weigh out, you know, precise amounts, you know, of water, you mix everything together, and every time you make up that media, it's going to be made the exact same way, okay? So in that situation, we're dealing with, you know, defined media, precisely known components of the media. So in the table, you know, you're seeing examples of what we have in a broth. So we have peptone, we have yeast extract, we have sodium chloride, right? So in the peptone, and in particular in the yeast extract, you know, things can change. LB broth and LB agar, you know, mainstays of, you know, labs where you have, you know, molecular biology being done with growing E. coli and things like this. They make tons of LB media, you know, both plates and tubes, you know, slants, whatever. And so, you know, we use this all the time, knowing that, you know, we don't know exactly what's in there all the time, but the cells will grow. On the other hand, we have, you know, minimal salts media shown you on the right side over there, and you see that you know exactly how much glucose, you know, how much, you know, sodium phosphate, etc., all the way down. You know exactly how much concentration of each you have in there. Okay, so there are times when, you know, it would be necessary to do that, and for most general growth of bacteria, right, we're on that side. Okay, we're using, you know, the nutrient agars, the LB agars, with the extracts and so on, and to grow the cells. We have specialized media, and so we can have media that's selective, we can have media that's differential, and as well we can have, you know, media that's highly enriched, okay, to allow for the growth of cells. And so if we have selective media, we can isolate, you know, microbes with specific properties. So which means in that particular media, you know, we, you know, would enhance the salt concentration or particular sugars like, you know, mannitol, and only cells that can utilize those, that can, you know, withstand those, will grow in there. Other cells cannot grow in there. We have, you know, differential media that will allow organisms to be recognized based on the ways that their colonies will appear. And so, you know, this allows you, upon visual inspection of the plate, and you see those particular colonies, you know what you have growing in there. And so many of those are based on, you know, on sugar fermentations like lactose. And one of, you know, really plate that's used in many microbiology labs is the McConkey agar. And again, you know, this will allow you to differentiate things, you know, like E. coli and so on from other gram-negative organisms. Enriched media, you know, can be used to increase particular populations of microbes, right? And so again, you know, the example given over here is for saboros, for fungi, but you know, we can use bacterial examples. You know, so for example, we have Neisseria is very fastidious. It needs, you know, a lot of enrichment in the media for growth, and so we have things like chocolate agar and things of this nature, you know, blood agar, things that, you know, are highly enriched that will allow certain organisms to grow on them. So again, specialized media, okay, examples being selective media, differential media, and these enriched media. And again, you know, fastidious organisms that are very difficult to grow, you know, typically the media will be enriched, you know, to allow for their growth to occur. And so we see examples of those over here. So, we have what we mentioned, the McConkey agar, which you see down at the bottom, right? And so you have growth there for, things that can ferment, lactose in there. And you see, an example of the EMB agar, you see methylene blue, right? And then we have brilliant green agar on the top, and you see the examples of organisms, that you can have growing in there. And so again, the thing is to remember these specialized types of media. And so when we talked about, the fact that we have, all these bacteria that we cannot grow, right? So we can identify them molecularly. We can do PCR, with the 6-ness, small subunit RNA, but we don't know what they need for growth. Okay, so what that tells you is that, when you identify, a source where you have bacteria, the go-to types of media would be the things that we've talked about. So you put them on things that we already know. And if you don't have them growing on those, then you're dealing with a situation where the components that we need, for their growth, are just unknown. Okay? And so you have people, actively, investigating, media, trying to understand ways that, these organisms can be grown. It's one thing to know that the DNA is there, but you can't describe what the organism looks like until you can actually grow it. And so again, we're seeing examples of these over here showing you what, the lactase positive situation looks like, where you can actually see the growth, the lactose negative situation looks like, and then where you have, a gram-positive organism trying to grow in, McConkey, and you see that, the growth is inhibited. So what we need to remember for our purposes here, okay, is that, right, so if we, have something that's selected for the gram-negative cells, okay, and we put, the gram-positives in there, right, we're inhibiting that. So that the fermentation of the lactose, which is what's used, for those that are positive, right, we can't even do that. The support of the growth just itself without fermenting the lactose, right, we can't do that if we're gram-positive. So we absolutely have no growth, okay? And, again, we talked about blood agar, and we saw the hemolysin. So right now, I think everybody can recognize beta hemolysin, alpha hemolysin, no hemolysis, right? Okay, so again, the type of media is used, to be able to identify these cells, because the hemolysin will hemolyze, the red cells for us to be able to see that. And so you can see the zones of hemolysis around the cells, and if it's clear, then we have beta. If it's, partial, greenish, we have the alpha and so on. So the solid media is good, okay, because, you have a substrate that you can actually put the cells on, right? And like we saw, they can grow and you can see the colonies. And so in those situations, where we started to discuss yesterday, which we'll see on Monday, where you're trying to make that decision, should I culture this thing or not? Should I try to see if anything is going to grow, right? If you, have, material from the plaque, for example, and you suspend it, in saline, PBS, right? You can put it on the surface of the plate. And, if you have stuff in there for which the growth conditions are right, they will grow. And you can see what the colonies are to describe them. And so we can separate the organisms, we can see the different colony types and so on, and allows us to know what we have in there growing. And so the common ways of doing this would be a streak plate, spread plate, or pour plate. The streak plate is extremely common because you're using a bacterial loop, and you can pick up, your cells, and you can, streak the plate, right? Pour plates, you mix them in with melted agar, and you pour that on your plate. And then, the spread plate, you have, these little devices, the glass ones, we always call hockey sticks, and you can spread, your cells on the surface of the plate. So each one of these methods will allow you to see, the bacterial colonies. The best of them would be the streak plate for you to see the colonies. The pour plate, your colonies can be embedded, inside the agar, okay? But these are the three, basic routine methods that are used for getting cells onto your plate. Go ahead.
Repeating what spread plate was?
So the spread plate, what you have, you know, they have, you know, different people manufacture different things nowadays, but the traditional way is you had the bent glass rod, okay, which is why they call it the hockey stick. And so essentially, you know, you put your cells on the plate and you use that device and you spread the cells on the surface of the plate. And then you incubate them and you wait for growth, right? So this example showing over here, and you know, I've used this slide, and I always, every time, you know, I've always, you know, mentioned that typically you work with 4 quadrants instead of 3, okay? And so, you know, if you want to obtain a pure culture, so you know you have the bacteria there, you want to, you know, separate it from everything else that's growing, you know, you do the streak plate and you do what's called streaking for isolation. So you want to be able to isolate, you know, the colonies away from each other. And so the purpose of having that, you know, 3rd or 4th portion of the streak plate is that that's where you're going to have your most separation, okay? And so if something grows on there, as you see over here, then you can come back and you can pick that one colony out, and then you can grow it further in, you know, in another dish. And so based on that, you can now either put it in blood culture or whatever, and you can do all the different studies that we're talking about. If you want to isolate DNA, if you want to do biochemical tests, whatever, you know, you have that pure colony now to work with. And so this is, again, a very basic way of doing things. And so we see an example of a pour plate. You mix this bacterial suspension with the melted agar, and then you pour it out on the plate. And so again, you know, you can see colonies on the surface, as well you can see colonies, you know, underneath the agar itself. And then the spread plate, okay, there's the bent, you know, glass rod, okay? And that's the one that's up here, right? The bent glass rod. And so you put your cells on there, and then you spread the cells out on the surface of the plate. So again, you know, different ways of obtaining pure culture. And so we emphasize this point, you know, what if the cells can't grow? We already said that. If they can't grow, then we need to use molecular means. And so we had the one schema that I asked you to please know the whole thing, right? So how you get from the, you know, the source of the cells to obtaining the DNA, to doing the PCR, right? And then, et cetera, getting the sequence, you know, of that DNA, and then being able to, you know, to do a blast search and so on. So on the kind of questions that you would get, it would be, you know, do you recognize the proper sequence of events? Okay, so how do you get from the source of cells to your blast search, right? So we don't want to mix up the steps in between. Okay, so that's why we wanted to know that. So we see different ways over here. So we can do the PCR. We can also label DNA sequences, you know, get probes from those, and then we can do fish analysis. And so you know, we need to know that term, fluorescent in situ hybridization. So we can, you know, label those probes with fluorochromes, and we can use that to find, you know, where cells are located. And so again, this just kind of shows you, you know, the scheme, how that would work. Okay, and so because it's fluorescent, you know, we're going to look under the fluorescent microscope to be able to identify things. And we can use multiple fluorochromes, multiple colors of the fluorochromes to be able to identify, you know, different cells in there based on the hybridization that we get with the probe. Okay, so when you see these, you know, with the questions, you want to recognize them as to what they are. And this is an example of that. So if you were shown, you know, an image like this, and you were asked, you know, how this was obtained, you want to be able to remember that, you know, this is through fish analysis, and especially if we're using DNA probes, so that we don't confuse it with, you know, something like, you know, confocal images from immunofluorescence, where you also have fluorochromes, but you're using antibodies, okay, in that case. Okay, does that make sense? So we don't want to confuse the two. Okay, all right. So again, fluorochromes. Right. So these are the different colors. The different. They like give you the different fluorescent colors. Right. Okay.
And what did we not want to confuse it with?
With immunofluorescence, you know, where we're using antibodies. So immuno tells you we have antibodies there. Right. Mm-hmm. Okay. Any questions about that? Okay, all right. All right, so obligate anaerobes. Which ones, A, B, C, or D?
See.
C. Okay? So obligate anaerobes, okay, no oxygen. Okay? Just like obligate aerobes must have oxygen. Okay? All right. So, we talked about metagenomics, okay? And so the only thing that I want to emphasize over here is the last point, okay? So, as much as it's, nice to have the DNA information as we've been saying, right? We still need to be able to grow these things. And there's been some progress made, in this area where organisms, that were identified using DNA methods, 10, 15 years ago kind of a thing. Right now, they can be grown, you can actually see what they look like. And so, and this is, of real importance in the population of cells that we have in the oral cavity, because one of the phylum, that we're going to be talking about, the TM7, right, is one that, again, because of its significance, there was a lot of effort to really try to morphologically identify this thing, because it's present in the oral cavity, right? So, not in the abundance level, like the 4 phyla that we've been talking about, but nonetheless it's there. And so, we'll say a little bit about it, and see some of the images from it. But so there's still, a need, to grow these things. And so being able to understand, growth requirements, media composition, all of these things, active areas of research, and in particular with the human microbiome, not just environmental organisms, but the human microbiome in particular in the oral cavity. This is a really robust area of research to try to find out what the population of cells are that we have growing in the mouth. And so again, within the environment, it's of importance too. But the point to make over here is that one of the difficulties that people encounter when they try to culture cells that are isolated, even from the oral cavity, GI tract, etc., is that these organisms, maybe cannot grow by themselves. Does that make sense? And so the TM7 phylum I keep talking about, because we're going to say much more about it next week, is one of those, right? And so what would be the benefit of knowing interacting organisms, that needed for growth? What would be the benefit of that?
And control the growth.
Right? So if you can control the growth of the required partner, you will kill the other member. Does that make sense? And so the strategy is just that. So when the TM7 phylum was described and found that, that the organisms had this need, to be with others, right? So the thing is, okay, if you know what those others are, and if you can get rid of them, then the main organism cannot survive. Okay? And so this is a very important part. So when we're trying to isolate organisms, we're so focused on just wanting the one organism. Does that make sense? So remember how we talked about isolating, the organisms, you're trying to do pure culture. So you're looking for one. But the strategy might have to change if you know that there are other growth partners. Okay? And then that allows you actually to be able to make the identification. Okay? So again, they may be accustomed to just being with their friends, etc., their neighbors, to be isolated, and so you have a hard time trying to just pull out one organism. Okay? And we have examples of this, within the oral cavity, with this TM7 phylum, and so on. So we can measure how much growth we have. We can count, the organisms. We can do, viable cell counting, etc. Turbidity using a spectrophotometer. We can do direct counts. So we'll look at one more slide and we'll take a break over here. So we can use, the common counting chamber that you have of counting bacteria is the Petra-Pauser chamber. It's, it looks very much like a hemocytometer. Has anybody seen what those look like before? Anybody seen one of them? Okay. So yeah, so there's little chambers on the slides, and they're dual chambers. You have them on either side, so you can load the cells under a cover slip. You put it under the microscope, and you can count the cells. So it can be done, very quickly, in the lab. And so the important thing about this is that when you're counting both live cells and dead cells. Okay? So the numbers you get might be inflated because, again, you're seeing both live and dead. You can count living cells. And so here you obtain the cells from your culture. You do a dilution of them. So typically you're doing, serial dilutions, log level. So, 1 to 10 type dilutions, 1 to 100, etc. And then you plate the cells out. Then now you count the colonies. And so what you're obtaining are called colony-forming units. Okay? So in order for you to form a colony, right, the cell has to be alive. It has to be divided. Okay? And then once you count the colonies, you multiply that back by the dilution factor that you made from your tubes, and then you get the cell number. So the important thing over here is that, when we get the colonies on the plates, okay, if we describe them as either being too many to count when you can't make out. So if you're counting and you're counting more than, one cell more than 2 times or whatever, that's too many, okay? And so, you want the cells to be between, 30 and 300, somewhere in there, where you can easily count and separate the colonies you're counting, okay? So those are countable plate, will be that. And then, if you're way out here when things are too dilute, okay, right, too few. Now, if it's a situation where, the cells are really tough to grow, you can't, it takes a lot to get, enough of the colonies on a plate, after you do the experiment, then, you're just happy you can see something growing, and then you can increase the number of those cells until you can do a proper enumeration. You can also, do, these 4 plates, and again, look for the colonies. There, you can filter them, and concentrate them, right, so that you can get enough cells to see when you don't have enough growing on the plate. Okay, so we'll take a break over here, and 5 minutes, we can stretch, and then we'll continue. Okay. Any questions? So did those make sense, for the note taking, right? Okay. That if we do direct count, with the counting chambers, the Petra-Pauser, you're counting live and dead. If you do the viability counting, you're counting live cells because you're counting colonies, and so we know countable plates between 30 and 300 colonies is where you want to be. And in some cases, if you have low concentration of cells, you can filter them to concentrate them. You can plate them out again to increase the number of cells that you have, and then you can repeat the counting and so on. And so we can also use the spectrophotometer to measure the cell density that we have. And so when we do that, we're measuring based on the turbidity that the cells produce when they're growing. So in the media, if you just put the clear agar in the spectrophotometer, you're going to get 100% light transmission, light transmittance through the media. But then as the cells are growing, right, and the media is becoming more turbid, right, the transmittance of the light gets reduced, and to the point where, you might end up, depending on how turbid the culture is, close to 0. And so we're measuring, the absorbance of that light by the cells growing in there. So that's the optical density that I referred to. And so again here, when you're doing that measurement, cells that are alive or dead, you're still going to measure the OD. Okay, so just like the direct counting on the Petra-Pauser chambers, right, you're going to be measuring, both live and dead cells present. And so typically doing the absorbance reading is accompanied by some other method, like the viability assay that we saw, so that you can actually count number of colonies that are being produced. So doing the OD readings, gives you an approximate measure of how much cells you're dealing with. And so in situations where you can't stop and do a count or do, a colony plate or whatever, and you're just wanting to know, do I have enough cells to do a particular assay, measuring the OD, will be sufficient, to give you that information. So we need to know this growth curve. We need to be able to recognize portions of it. So is there anyone that's never seen this before? Growth curve, everybody's seen this before, okay? So we know the parts of it. So, when we put cells into grow, usually, we have a period of time where the cells are essentially preparing to divide and grow. We have a lag phase. And the lag phase, depending on the bacterial type you're growing, can be long, okay? And then, the cells take off, they begin to divide. So again, the general example, like E. coli every 15 minutes. Of course, this would vary depending on the type of cell that you're growing. Some might take a little longer. And so you have the exponential phase, and then as nutrients begin to get depleted, cells, begin to die. We get the stationary phase, and then we have a decline or death phase. Okay, so this is a very important, thing to have when you're working, especially with cells that you're not very familiar with. You don't know. You're trying to determine, the behavior of the cell, in culture and so on. Then this is the kind of a thing that you would need to do, and to kind of get a sense of what you have in here. And so, if you wanted to kind of get a clear sense of the cell numbers that you have, so at each of the time points, you can actually do a viable cell count, and you can count the colonies that you have, and you can see if things are decreasing, which is how we can determine if we're in the stationary phase going into the decline or death phase. So this is a typical bacterial growth curve, okay? And so we need to know the parts of it. And so, yeah, go ahead.
During the stationary phase, is there a combination of cell death and cell growth, but it's staying stagnant because the line
it's
is the
getting
same?
it plateaus, yeah. And so, so the thing about this is just, again, the way you would have a question like this, would be making sure that we know what's going on in each part of this, graph. And so, with the growth curve, things that we can, know, and things that we can kind of evaluate, the generation time, okay, so doubling time of the cells, the growth rate, the yield, of the cells and so on. And so all of this can be obtained. So part of, doing the viability as you go along, will allow you to get this kind of information, okay? And so depending on what you're doing, this could be pretty important. For most cases, it's just that you want to know the behavior of the cells that you're dealing with. And so we can do continuous culture, so some situations have a need for this. Most people will not do a continuous culture. And so again, if you want to keep the cells at a point where, they're pretty much, in exponential phase all the time, you want large numbers of those cells to work with, right? You can, essentially have the waste, removed from the media, replenish, fresh media, so the cells don't enter the stationary phase, okay, as quickly. Does that make sense? And so you can have things pretty much, in that log phase where, metabolic activity and all of those different things, are at their peak. And so that would be an important reason, to have that. And so, the critical thing about being able to maintain these is making sure that you have enough of the waste removed and enough replenishment, with the fresh media to keep, the nutrient levels pretty stable to allow you to have that exponential growth in there. So again, we can kind of give you an example. There are different permutations of this, that people would have in various labs where they have continuous cultures going on. And so again, the key thing, is again having those nutrients available, having the waste removed, and as you're removing the waste, you're also kind of trimming down the number of cells you have in there, so that you can maintain things, within that log phase. And so in terms of the microbiome, you know, in the human body, you know, one of the locations that's thought of as a continuous culture is in the GI tract, okay? Because, you know, many of the things that we just talked about kind of, you know, fit that. You know, nutrient levels, kind of decrease, you know, in the abundance of the cell number, and always kind of maintaining, you know, the population of cells at this, if you will, you know, a homeostatic level that allows for different aspects of the host to function properly. So the GI tract is one of those. Okay, so this next area over here, we just kind of want to look at this, and so when we talk about these pathogenic states, you know, next week, you know, we're also going to talk about, you know, situations where we want to prevent growth, and how do we do that? So physical, you know, type of things, chemical, and so on. And so we want to be able to know, you know, what disinfectants, etc., you know, do, you know, how we use heat, you know, the categories of antimicrobial drugs, how they're tested, and you know, which ones impact gram-negative cells, gram-positive cells, and so on. And so, you know, the targets of antimicrobial therapy, you know, are we targeting the cell wall? Are we targeting, you know, DNA replication? Are we targeting, you know, RNA, you know, transcription, and all of this? So this would be, you know, part of number 4. We have a couple of tables where I'm going to tell you the same thing. You know, we want to know, okay, which ones are going after peptidoglycan, which ones are going after, you know, DNA replication. We want to make sure that we can recognize those. The second thing, you know, for this class that's going to be important, so if we're looking at the specificity of the antimicrobial agents, which ones have broad, you know, applications, which ones are very narrow, only mostly gram-positive, mostly gram-negative, and so on. So those are the kind of things that we want to kind of keep in mind as we're going through this. And then, you know, how is it that we get, you know, drug resistance to occur? And so everything, you know, from, you know, the participation of, you know, things like the phage or the plasmids that we have within the bacteria, right, you know, to the environments in which we have the cells growing. So we have, you know, the question over here, how can we eliminate microbes or inhibit their growth? So we just finished talking about how we want the cells to grow, right? And, you know, why it is that we need to culture, right? And so then now here we're talking about how do we want to eliminate or inhibit the growth. Okay, so on the one hand, right, we would, you know, if we wanted to investigate, you know, particular organisms, we would like to grow them. But if we have pathogenic states that are being contributed by the organisms being present, then we want to either reduce the numbers very drastically or eliminate them altogether, right? Again, going back to the symbionts, pathobionts, you know, opportunistic organisms that we talked about. So we need to know what's needed, you know, to do that. So we can remove things physically, so we can filter, you know, various sources to get rid of bacteria. And, you know, there are many ways of doing this. If we have situations, you know, where the agent, you know, that contains the bacteria will be destroyed by heating or by autoclaving, then, you know, we can filter, okay? Or in some cases, you know, we can use gases, you know, to sterilize. And so the thing we need to remember right here is that the typical pore size that's used for filtering to eliminate bacteria, you know, between 0.2, 0.45 microns. 0.2 very common. Okay, we can eliminate, you know, some of the smaller bacteria. But if we needed to get, you know, things like mycoplasma and rickettsia, so very small, you know, chlamydia, those type of sizes, we might need to do ultrafiltration, just like we would do for viruses, you know, to get rid of them. And so filters can clog. So, you know, if we're trying to filter large amounts of material, you know, that might not be the best thing to do. If we're trying to do ultrafiltration, we need high pressure. And depending on what you have in the media, if it's very thick, you know, if you have, you know, numerous macromolecules in that, you know, so your solution of the media, then, you know, you're going to clog your filters pretty quickly, okay? And so again, 0.2 is very common. So, you know, a person is working, you know, it's a tissue type situation, where you want to eliminate bacteria, 0.2 is the most common type of filter that's going to be used. For most general things, 0.45, okay, but again, 0.2 pretty common. So when we're filtering, we can reduce the microbial population, and if we have pretty much, if you will, a homogeneous size of the organisms, you can sterilize the media that way, okay? You can sterilize the solution that way, and you don't have to heat up the media. And, if you have organisms that you have, contaminated air, you can also filter, okay, to reduce or eliminate those organisms. And so pretty much this kind of just shows you, what kind of things look like. So you can have the organisms, on the filter, you can retard, them from going through. And, in the same way that we mentioned that you can concentrate the cells using the filters, right? The reason why you can do that is because when you concentrate them, they'll stay on the filter, on the top part, and then you can actually take those and you can actually grow them. So we're trying to, we're trying to eliminate them, right? So we filter, then this part, gets discarded, and we can autoclave it, put it in the biohazard, container and get rid of the cells that way. So again, filtration is pretty important for that kind of a thing. So again, we're looking at the membrane filters. We have the porous membranes, have a pore size, and then you can screen and do so. We can also filter air. So again, masks, cotton plugs that sometimes you can see on vessels. We can, for hoods, we have the high efficiency particulate air filters or HEPA filters that can actually, remove bacteria. And so this is again, in safety cabinets, you have the HEPA filters. They're very important, and they will prevent cells, bacteria from, falling onto the surfaces or on the media that you're working with, and as well, protection for the person who's working, on the outside of the hood. So that's also important. We can use moist heat. We can, destroy, bacteria, fungi, etc. We can boil, etc., but we cannot kill endospores. So use of moist heat alone will not kill endospores. So that's the most important take-home message that we're trying to get here, okay? So when we do the heating, we can denature protein, we can, affect, nucleic acids and so on, cell membranes, again, but we cannot destroy endospores, okay? We can't do that. And so what this is showing you is that, again, with yeasts, right, molds or fungi, right, we can, have, destruction if we go all the way up to 80 degrees, Celsius and so on, bacteria, go all the way over there, but, endospores, will still be a problem. So we need the heat, we can manipulate the temperatures, and we can also provide pressure that would allow those endospores to be killed. And so when we see the 100 degrees centigrade over here, vegetative cells, okay? So we're distinguishing between vegetative cells and endospores, okay? So we just want to make sure that that's clear. And so when we use the autoclaves, we have pressure in addition, to the moisture, and then we also have the temperature.
So what is the difference between vegetative cells and endospores?
So the vegetative cell, doesn't have, the spore coat and, all the factors, that the endospore has that makes it so resistant. So the vegetative cell is the, the plasma membrane, covering the cells, cytoplasmic contents, etc., right? Those, you can easily kill. But if they have endospores, right, that's where the concern comes in, okay? And so we see the point, over here. If you have, these archaea, the hyperthermophils, right, they can live at 100 degrees, so 100 degrees is not going to kill them, okay? You have to do more, okay? And so for those kind of organisms, you know, you can up the temperature, you know, increase the amount of time things are in, you can get them. But again, they're archaea, but they're still vegetative cells. Okay? All right. And then if you cannot heat stuff, right, then we need to use things like gases and so on. So the autoclave, you know, is really important. Now, one of the things, you know, whenever I do this class here, same thing, you know, when I teach, you know, a micro at the cleaning state, this part, you know, if we forget everything else, right, and if we can remember gram stain and the need for the autoclave, we're okay. Okay? All right. Those two things. Okay, and that's good. And this is because, you know, when you're autoclaving things, you're trying to sterilize, you might have equipment, you know, you have instruments, you have media, you have, you know, fabric, you know, dressings, things that are going to be in contact with tissues and so on, externally and internally. And so you want to make sure that these endospores are killed. And so the use of autoclaves in institutions is highly monitored. And this is because, you know, we want to make sure that the autoclave is working, okay? And so we have organisms that themselves, okay, produce spores that are used as the indicator for the effectiveness of the cycles, okay? And so if you're going to autoclave, you know, biohazardous materials where you have a lot of bacteria, you know, you have endospore formers, then you will put that stuff in the environment, EPA wants to know that the autoclave cycles work. Does that make sense? And so these organisms, you know, are the indicator. So they're put in the autoclave for the cycle, and they come in different, you know, configurations. You know, the most common ones is that you have, like, you know, ampules. On one side you have the media, on one side you have the organisms. At the end of the autoclave cycle, you kind of break the connection between the two so that the media and the cells mix, and then you incubate them. So you incubate them, and okay, you check 24 hours, check up to 48 hours, okay, in some cases even up to 72. If you see growth, then the cycle failed, right? Because if the cycle worked, those organisms, the spores should be gone, okay? So it's a very important part because, you know, as you know, with what you're doing, you know, you're going to be having things that need to be autoclaved, you know, your instruments and everything, and those are regulated. And the autoclaves come in different sizes and different, you know, permutations and so on. And so the thing is to make sure that that piece of equipment is working the way it should work. Okay. All right. So endospores, endospores, endospores. That's what we want to get rid of. And that's what the autoclave is helping to do. Other kinds of things, you know, pasteurization, right? So again, you know, temperatures, you know, high, but, you know, below boiling. And again, this is used to, you know, reduce bacterial load. So we're not eliminating 100% every single bacterial organism that's in there. So we're not sterilizing, right? We're just reducing, you know, the population load. We can do dry heat, okay? So, you know, it's effective for some things, but, you know, moist heat, you know, is better in most cases. And long exposure times, you know, high temperatures and so on. So whereas with the autoclave, you know, the standard, you know, 15 minutes, you know, 121 degrees, you know, at high pressure, you know, 15 pounds of pressure. That's, you know, at minimum. So if you have, you know, a packed autoclave, and if you have things, you know, where you can't get the you know, the steam to penetrate very easily at 15 minutes, then you're going to increase the amount of time and so on, right? But, you know, 121 degrees Celsius, 15 minutes, 15 pounds of pressure is about, you know, where we are to make sure that we're killing, you know, endospores. And so, you know, we've talked about heat, moist heat with pressure, and so, you know, we can have, you know, freezing for preserving essentially the cells. And so typically the cells mixed, you know, with some glycerol-based solution to prevent ice crystals from forming inside the cells and in the membranes, which will kill them. But then, you know, if they're in this glycerol mix, you know, dimethyl sulfoxide, then, you know, at very low temperatures that we have for liquid nitrogen, we can still keep, you know, the cells alive, and we can bring them out from that, and we can plate them out of media, and they will grow, okay? So that's what that's showing. UV radiation is also important, and so we can damage DNA, you know, with this. And so we form these thymine dimers. You know, typically cells will try to repair if you have the dimers being formed, but you know, depending on the length of exposure, you have so much damage going on that the cell cannot keep up with the repair, and you know, this is how you would kill them. So most biosafety hoods, for example, will have a UV bulb in them. So you turn that on, you know, let the UV, you know, radiation hit the surfaces and whatever is inside, and then, you know, you can now utilize that. And so you can see that that's what's being used to kill cells, you know, in this water that you see with the school children. In any case, we can use disinfectants, we can use antiseptics, and again, we need to know, you know, we tend to use them interchangeably, but they're not really the same thing. So disinfectants you can use on non-living surfaces, you know, on fomites, you know, doorknobs, that kind of a thing. Antiseptics, you know, the things that you can apply on tissue, okay, living tissue. And, you know, typically on the outside of the body, you know, to reduce bacterial growth. And so there are a number of features that are important if you're trying to have an antimicrobial agent. You want something that can kill a broad range of organisms. You know, you want the thing not to be corrosive or have, you know, be toxic or have a very bad odor. So there are some, you know, real, you know, really effective antiseptics, you know, that have been made. But some of them, you know, the smell is so bad that, you know, people who have to work with them have a real difficulty, okay? And so you find people not using them, okay? And so you don't want it to leave a residue. You want to be able to wipe it up, you know, so if you have it, you know, around, you know, areas where you're working, you want to make sure that there's no residue, no fumes, and it should be reasonable in cost, and you want something that's pretty stable, okay, stable shelf life, okay? All of these things are thought about when stuff's manufactured. So on this over here is just to look at examples, and we see that, you know, one of the most common items that's used to, you know, clean surfaces and get rid of organisms is ethanol. So 70% ethanol. And, you know, so if you compare 70% ethanol with something like 95% ethanol, the more water you have, which is why the 70 is good, the more absorptivity you have to give the alcohol enough time to do the killing. So we have that. So 70% ethanol is used a lot. And so, you know, you have it in many of the things that people buy for, like hand sanitizers, whatever, it's based on that. And then, you know, phenol, okay, and the derivatives of phenol are also important. Things that, you know, do oxidation, you know, sodium hydrochloride, bleach, things of that nature, all the way to things that do cross-linking, like, you know, glutaraldehyde-based type things used for antimicrobials. Practical type of things, you know, to decide how you pick something that works. A lot goes into doing that, depending on what it is you're trying to sanitize or reduce the bacterial numbers for. So all of those things, you know, play a role. The last point on here, they're pretty self-explanatory, and the last point over here, you know, is that yes, we're trying to eliminate, you know, bacteria, harmful, you know, ones that potentially be opportunistic. But, you know, how toxic are some of these things, you know, to our own cells? That's always a question, and you know there's ongoing debate about that, okay, you know, about the various reagents that are used to make sure that we're not harming ourselves as we try to use these antimicrobial agents. Before an antiseptic or disinfectant, you know, or any other antimicrobial agent for that matter is used, right? We have to know how effective it is. Okay, so you want something that's effective, short period of time, kill the most number of cells. Okay, so that's really what you're looking for. And so, and I only put this over here just so that we kind of know that. So if you're measuring effectiveness, right, we have all different types of factors, one of them being, this decimal reduction time, right? And so here you're targeting 90%, how much time does it take to get 90% of the cells killed, okay? And pretty much, just under regular conditions, and you can vary the conditions, okay, depending on moisture level, the number of, items that are in contact with the bacteria that need to be, killed, and so those can be worked out, to find what that particular level is that's important. Drugs that are also used, okay, how do they work? So we have antimicrobial drugs. These are different from antiseptics and disinfectants. And so, we have these, they're not topical, things that are going to be taken internally. So the things that we have to remember with the bacteria, we have gram-positive cells, we have gram-negative cells. Sometimes we have agents that cannot kill both. Ones that are, directed towards gram-positives, one towards gram-negatives. And so we need to be able to know that. So whenever antibiotics, disinfectants, antiseptics, when they're evaluated for both cell types, for gram-positives and gram-negatives, okay, to see if we have a broad effect or if it's very specific. And if we have it, effective, for gram-positives, we want to make sure that it'll kill pretty much all gram-positives, right? And so what may work on gram-positive and gram-negative cells may not work on acid-fast cells, right? So, again, all of those things are taken into consideration. And so again, if you notice the point over here, right? You eliminate one class, and you have others that you can't do anything about. And so, this is just, you can look at this on your own later on. It just kind of shows you a timeline of, the use of, the various antimicrobial agents, all the way, from penicillin, etc., the development of resistance, all the way to where we have now multi-resistant organisms, okay, to many of the antibiotics, that were very effective, we have organisms now that can be, resistant against all of those. And so there's a big concern for that. And so this just kind of shows you, again, the table, and I'm not going to ask you specific questions about that, except for the one chart we're going to look at, where we see, which of the agents have broad activity and which ones have more narrow. But it kind of gives you an appreciation for, the different, natural antibiotics, that have been in use, and then the derivatives of many of those, where we have, semi-synthetic, and in some cases, fully synthetic types. And there's always need for more because of this resistance that we have developing. So clinically, especially, within hospitals, we have, Clostridium difficile, problems with that. We have, multi-resistant, staph, and most recently, and, we don't know how bad this is going to get, we have Candida auris, which is a big concern to, to many because, it's being found more and more in places. And so drugs that, were once effective are no longer effective. And with this particular organism, there hasn't been a clear explanation for how it got into the hospitals to begin with. Transcription, folic acid synthesis, ribosome function, we have, drugs that target those. And with the penicillins, we want to remember what the major component is. And that is this beta-lactam ring, okay, that we have. And so because of the effectiveness of these penicillins and the derivatives that have the beta-lactam ring, so remember we mentioned in the bacteria and gram-negative cells, have this, but so we have, the enzymes that are produced that attack the ring, okay? And so, this is an important thing for resistance, in bacteria, being able to target, the areas of the drugs, where we have the activity, okay? So, number one, we want to know what are some of these targets, and we have them listed. The first one always is that peptidoglycan, because that's the biggest thing outside of the cell. If you can not form it, then the cells are going to have a hard time, developing and growing, and so on. So even for, gram-negative cells that have a very thin, peptidoglycan, you still kind of want to go after that, okay? And it's a little bit more difficult because of the outer membrane, right, that you have, to get the drugs, in. And so gram-negative cells will spit out the drugs, okay? They have all those porins, and so they can resist, when the drugs are available, it gets inside, they'll extrude them. And so they can resist that way, okay? And so this particular figure over here, okay, as we're finishing up, we want to kind of know. So we're looking at the different groups of organisms. So we have our gram-positive cells, gram-negative cells, mycobacteria. We have, the chlamydias, we have the rickettsias, right? These other, cells, that, again, variability in the cell wall. And so what we're looking at, before we begin to look specifically at the targets, so we have, things like the penicillin, so the natural penicillin, before we had all the derivatives and so on with the semi-synthetic. Again, very, narrow to the gram-positive cells, again, because of that thick peptidoglycan, okay? Then, we have, for the gram-negative cells, we have the semi-synthetic penicillins that essentially can affect, some gram-negative cells, some chlamydia, right? And then we have, the cephalosporins, the sulfonamides, again, gram-positive, gram-negative, okay? And then, we have, ones that can affect the gram-negative cells as well as the mycobacteria. So remember, the mycobacteria are acid-fast, okay? They have the waxy cell wall. And so the tobramycin, can affect, gram-negative mycobacteria, but if you look all the way down, right, we have the adenosine, very direct for these acid-fast cells, okay? And so streptomycin, again, mycobacteria, gram-negative cells, but the one that has the broadest reach is the tetracyclines. I mean, so broad, in fact, that, tetracyclines, they're used as prophylactics for epicomplexans, in particular Plasmodium, for malaria. And for the longest time, nobody really knew why it was working. And, so now we know the Plasmodium have an epicoplast. The epicoplast is derived from, photosynthetic, cell, which is no longer active, so of bacterial origin. And so, it's still an essential component for the cell. So if you, destroy the epicoplast, or if you prevented, molecules that are being transported into that, you also affect the Plasmodium itself. And so it kind of makes sense that something like this, with this broad reach, has that effect there on these eukaryotic organisms, okay, because of that plastic that's in there. So again, as we look at this, we want to kind of, remember this distribution that we're seeing with the effectiveness of these groups of drugs. And we'll stop with this slide over here. And so we're just kind of looking at, the drugs themselves to see what it is, that is most important. So we already mentioned, the beta-lactam ring, right? And so we have the target, so the peptidoglycan transpeptidases, and so we have the natural penicillin G, and then the various, semi-synthetics, penicillins and so on. And so again, we can affect that peptidoglycan, and we can affect the bacteria. We also have, other things like bacitracin, which is used on this, on the skin, glycopeptides, and again, they're targeting the peptidoglycan. And so again, remember, you have the, as you're forming the peptidoglycan, you have the pentapeptide cross-link that you can have, you have the tetrapeptide and so on. So again, if you prevent those from forming, then you can't do the cross-link. So which means then, you're not going to have a very effective peptidoglycan cage on the outside. So anything that disrupts that will affect the cell, okay? And so again, it's important to kind of keep these in mind. And so when we come back next time, we'll finish up the last bit of stuff in here, and then we want to start looking at the pathogenic states in more detail. And so we know now, names of the bacteria, that we can find in places. We know which ones we can associate, with different pathogenic states. So then now we're going to look at examples. We're going to look at, for instance, the 8 types, see, what happens in each case, which organisms we have predominant, which ones we have interactive, and get a better picture now that we know how we culture, how we isolate, all of these different things. And kind of get a fuller picture of what's going on under each one of these pathogenic states. And then the last thing we're going to look at, which takes us into the following week, would be the systemic situation. What happens when these organisms, from the oral cavity get in the blood and then, get into different places under various conditions. So, we have things like, chronic diseases, type 2 diabetes, all of this. We're going to look and see why these conditions kind of make, the pathogenic state in the mouth worse, and situations where, the state in the mouth makes the chronic condition worse. We're going to look at that. And then we have some rare, conditions that, we don't usually see, regularly. And so we're going to also look at that. And again, these, polymicrobial in nature, and start out in the oral cavity and can end up with a lot of destruction around the face when there's no intervention. So the material that we've been kind of looking at up until now will allow us to talk about these different situations with terminology now that we can apply to those. Any questions? Okay, so I'll get those to you, and then this will be for next week.